So last time we kind of went through the functions of p53 and kind of seeing, I think you guys can now appreciate why it's so critical in terms of its function as a tumor suppressor, just because of all the broad spectrum effects that it has and are initiated by its function. So again, we've got probably at least a dozen, maybe closer to 24 other tumor suppressor genes now that have been identified. But p53, more than any of them, carries out the greatest number of functions in the greatest number of cells, and that's why it's so important. So remember that earlier on we were talking about how RAS is the most commonly altered oncogene that you're going to find associated with human cancers, okay? The same is true with p53 in that p53 is the most commonly mutated tumor suppressor gene that you find in the broadest number of human cancers, okay? So like we talked about to wrap up on Wednesday, the vast majority of mutations that affect p53, they're recessive, and we'll get into that in a couple of moments, but most of them are point mutations that occur within a defined region of the p53 coding region, and what they create are missense mutations that eliminate the ability of p53 to bind to DNA, or, and these are coupled with that, they make the protein very, very unstable, okay? So in essence, the mutated copy of p53 doesn't really contribute anything, okay? Now we're going to see in a minute you could actually have other mutations that have a different effect, but by and large, that summarizes most of the mutations that you find in p53, okay? And if you take a look at this chart up here that comes from Weinberg's cancer book, goes back to about 2014, you can actually see that there's a pretty broad spectrum and also a gradient of different types of human cancers that have had inactivating mutations in p53. So the one thing that you can see is not all of them are equal. Certain types of cancer are more susceptible to inactivating mutations in p53 than others, okay? But there's two up there that I put stars by because we're going to expand on this a little bit this morning, okay? And that is, if you take a look at near the top, okay, you look at head and neck cancers, which are basically oral and throat cancers, you notice that they actually have a relatively high proportion of inactivating mutations in p53. But that's historical. It's not current. And one of the things that you're going to see is that that type of cancer is on the rise. But the new types of cancers that are being diagnosed still have unaltered p53 genes, but they don't make any p53 protein, okay? And if we come all the way down to here, it turns out that cervical cancer, even though it has a very low incidence of inactivating mutations in p53, virtually all cervical cancers lack functional p53 protein. So there's two paradoxes up there that we're going to spend a little bit of time talking about, because what you're going to see is those types of tumors don't produce p53, but it's not because of mutations, it's due to a virus, okay? All right. Here's a trivial question that a lot of people don't appreciate, is that when p53 was first discovered back in the early 1980s, it was identified as an oncogene. It wasn't discovered as a tumor suppressor gene. And the reason for that is that the mutations that were first discovered in p53 behaved like a dominant oncogene, okay? And that is, the cells were more prone to proliferate when you had a copy of this mutant form of p53 expressed in heterozygous cells that had a wild-type copy of p53, okay? So what was going on there to explain this, okay? Well, again, like what we talked about on Wednesday, the vast majority of mutations that you find in p53 are recessive in their loss of function. So what it means is you can consider the protein being produced by the mutant allele, it's invisible, it's not there, because it can't contribute to the level of p53 protein. So what it means is that if you're heterozygous and you've got a wild-type p53 gene and you've got one of these loss-of-function mutant alleles, your cells are going to be able to produce half as much p53 compared to an individual who's homozygous with two wild-type copies of the p53 gene. Now remember, the effects of p53 are dose-dependent, okay? So that the ability of p53 to become stable, stop the cell cycle, activate repair enzymes, activate apoptosis, those are all contingent on the concentration of p53 your cells can accumulate in response to DNA damage. So if you at best can only make half as much p53, then what it means is that your cells are not going to be able to stop the cell cycle, activate repair genes, or activate apoptosis nearly to the same extent as a cell that has two wild-type copies of p53. So that's the situation with most p53 alleles. But this odd form that behaved like an oncogene was a dominant negative form. And the way that it works is you have to take into account that the functional form of p53 as a DNA binding protein is it is a tetramer, okay? So when p53 is phosphorylated, it becomes stabilized, it assembles into tetramers, and it's those tetramers that bind to DNA, okay? So this one particular type of dominant negative allele, a p53, it typically affects the transactivation domain. The transactivation domain is the region of the protein that recruits RNA polymerase and other transcription factors to the target gene after p53 binds, okay? So that mutant form of the protein can still bind DNA, but it won't do anything once it's there, okay? The problem is those subunits are going to be toxic. They don't work. But not only do they not work, if you incorporate, so let's say we've got a heterozygous cell that produces equal amounts of wild-type p53 protein and this dominant negative form of p53 that can't activate gene expression. So it's there, but it's useless. Well, if they're produced in equal amounts, and the possibility of any tetramer being made is completely random, then what you find is the vast majority of your p53 tetramers in this situation are not going to be functional. Because if you even have just one mutant subunit of p53 in a tetramer, it's dead. So you might think, well, half of them ought to work. Well, they don't. The fact is, 94% of the p53 in these cells is functionally inactive because the tetramers are dead. They're poisoned due to the toxic one. So you can see now in this particular instance, if you have this type of dominant negative allele of p53, your cells are never going to be able to mount any p53-dependent functions in response to DNA damage. So these cells are incapable of arresting the cell cycle, they can't activate DNA repair enzymes, and they're not going to be able to undergo apoptosis. All right? But this is why p53 was first identified as an oncogene, okay? It's not the prevalent form of the mutations that are out there. It just happened to be, serendipitously, one of the first ones that was discovered, okay? So just to let you know that you can get both recessive and dominant mutant alleles of p53. Okay, so this is just a little review article that came out earlier this fall that basically summarizes all of the approaches that are out there to basically deal with how can we correct or how can we repair or restore p53 function in tumor cells. The newest approaches that are being done to approach cancer then takes advantage of the fact that the p53 is being expressed. Okay, that's number one. Two is that the protein can't function because it's very unstable. Now remember, even wild-type p53 is very unstable. So the approach is, if you could come up with a chaperone-based therapy, a small molecule that can bind to p53, and it turns out p53 fortuitously, each of the monomers has a little cleft or a pocket that sort of nucleates the ability of it to fold into its native structure that's more stable. And so experimentally, it turns out this looks like a relatively promising approach because you can find small molecules that will function as chaperones. And what they can do is that they bind to this pocket on p53, and what it will do is stabilize it sufficiently to where now it can form tetramers, and it can now bind DNA and restore its functionality. So the idea is this, is it's an alternative way of thinking about how to go after and target a large number of tumors, not by going after the oncogene, not by having to use toxins, for example, like Taxol to kill the cell, but rather allow the p53 that can be present in those cells to let it handle the job of committing apoptosis. The beauty of it is that now it's cell autonomous, okay? It's only going to affect the cells which are expressing the mutant form of p53, which are tumorigenic, because only those cells are going to be able to accumulate p53 at a high enough level to where now they will be able to execute the apoptotic program. So, it's basically turning this whole issue of cell proliferation on its head, and basically instead of trying to murder it from the outside, you're trying to get the tumor cell to commit suicide, okay? Now, something that came out that was highlighted in this review article, it actually goes back to a finding that goes back a number of years, back to an epidemiologist in the 1970s by the name of Richard Pato. And Pato noticed that there was a striking lack, there was an interesting paradox, and that is not all multicellular organisms come down with cancer at the same rate. And that is certain organisms are less likely, far more less likely, to develop cancer than others. And the most stunning paradox that he observed were the larger the organism that you found, like African elephants, are bigger than we are, they have more cells than we do, they live for very, very long periods of time, so you would think that for African elephants, they should be as susceptible to cancer as we are. And it turns out they're not. So it turns out that the larger the animal, it turns out the less likely they are to develop cancer. So, 40 years go by without much of an explanation to try and figure out what's going on, what explains this. What actually took until 2014, 2015, that you had the completion of the Elephant Genome Project, and they went in and actually sequenced the African elephant genome, and interestingly, here's what they found, and this is the explanation. We all are diploid, so we're us lucky people. We have two p53 genes. You get one from mom, one from dad, that's it. One goes bad, you're at a higher risk for cancer. Two go bad, you're going to get cancer. African elephants, turns out, have 20 copies of p53. Not one, not two, they've got 20. Okay? So what does it mean is, these cells basically have backup on backup on backup. They can mount very, very, very rapid and very quick cell cycle arrest, DNA repair, apoptosis. You never have to worry about cancer to a certain extent, simply because they can mediate a much, much more robust DNA damage checkpoint because of all these extra copies of p53. Now, the interesting thing is, as part of this genome initiative, they actually went back and sequenced the genome of woolly mammoths that they were able to recover. And it turns out they have about a dozen to 15 copies of p53. So the take home from that is just because you have more p53 doesn't mean that you're necessarily guaranteed not going extinct. So anyway, this is kind of sad, but for a lot of reasons. Okay. So, Mutations are not the only way to take out p53. As it turns out, a number of tumor viruses out there also target p53 and other tumor suppressor genes, okay? A wide number of tumor viruses. But the one that I want to focus on in particular is human papillomavirus, okay? Because many tumor viruses have an oncogene. They'll have a given oncogene. Understand that the job of the virus is not to cause cancer. Okay? The agenda of the virus is not to cause cancer. The agenda of the virus is to create more cells for the virus to infect. Most tumor viruses can propagate themselves in infected cells one of two ways. They can infect what are called permissive cells, and what that means is the virus goes in, produces all of its proteins, it generates more infectious viral progeny that then bud off or secreted from the cells, or the cell might lyse, and that releases more viruses that can go out there and infect more cells. The other way that these viruses can propagate, particularly in what are called non-permissive cells, is that the viral genome will tuck itself and insert itself at a site within the infected cell's DNA. So now what it means is that the viral DNA sequences have to be duplicated or replicated and propagated during S phase and during mitosis when the infected cells divide. And depending on the circumstances, these viral DNA sequences can also go into a lytic mode of infection and allow progeny virus to be produced in response to certain stimuli. The point is this: viruses are parasites. The viruses require more cells to infect, irrespective of which way they go in terms of permissive or non-permissive infections, okay? A virus that can cause cells that it infects to proliferate has an evolutionary and selective advantage. It makes sense, because when that virus infects a cell, it can cause the infected cells to proliferate. It makes more cells. That's the reason why tumor viruses have oncogenes, okay? Most of them have one. Some of them have two. The master is human papillomavirus. Because human papillomavirus, there's different strains, we'll talk about them in a second, don't have one, don't have two, they've got three. So HPV has three viral genes that are oncogenic. Viral genes are often referred to, you might see this in general, as being prefaced with an E or an L. E means early. Early genes are genes that are expressed immediately when the virus infects the cell. Late genes are usually genes that make up the viral coat proteins or capsid proteins. So they're made after the virus has replicated its DNA. So anytime you see E in front of a number, for example, in the case of HPV, the three oncogenes are E7, E6, and E5. It means these are proteins that are expressed as soon as the virus enters the cell. If we take a look at what these three oncogenes do, it's interesting. E5 mimics a growth factor, so we'll take it out first, okay? So E5, when it's expressed in cells, actually goes through the secretory pathway, is put out into the bloodstream, and binds to PDGF, platelet-derived growth factor receptors, on the plasma membrane of other cells. Those cells will now behave like they're being exposed to a growth factor. It's stimulating them to proliferate, even though the virus isn't in those cells, okay? The other two are more insidious. HPV E7 binds to the retinoblastoma tumor suppressor and ties it up and takes it out of circulation. Now remember, what's the normal function of the RB gene? RB, remember that RB normally binds to a transcription factor called E2F. E2F is necessary for cells to pass the restriction point and to go into S phase and replicate their DNA. Our cells normally take out RB by phosphorylating it when we activate cyclin CDKs. That's what the growth factor pathway does. Well, HPV does it on its own. So HPV E7 binds to RB and displaces it from the E2F transcription factor. So HPV-infected cells now proliferate as though they were treated by a growth factor. Because now E2F is going to be constitutively active in those cells, and the cells are going to go past the restriction point into S phase because the virus uses the replication enzymes to copy its DNA the same way cells do to copy their own chromosomes. So here's one tumor suppressor that's now taken out of action. P53 is the next one. The E6 gene encoded by HPV is a ubiquitin ligase. It behaves just like MDM2. Only when the virus infects the cell, E6 is expressed at a very, very high level, much higher than MDM2. So now what happens is, in HPV-infected cells, your P53 levels are very, very low. They drop even further, okay? So the problem with HPV that makes it such an insidious tumor virus is that it takes out two of your tumor suppressor genes. Those are cell autonomous, so that's taking out the cell that it infects, makes them more prone to replicate their DNA. But now it also causes neighboring cells that can bind to the E5 protein. They will also proliferate. So HPV is particularly good at causing cells that it infects and neighboring cells to proliferate. The agenda being it makes more cells for the HPV to infect. Okay? So why is this particularly insidious to certain forms of cancer? Well, the easiest way to think about it is HPV is an STD. Okay? HPV is transmitted sexually. All right? It's without a question the single causative agent of 99% of all cervical cancer that's out there. Okay? So here's an interesting change if we look at this from an epidemiological perspective. If we look at certain cancers as a consequence of HPV diagnosis and HPV treatment. Okay? And what you find is we go all the way back about 30 years into the mid-'80s. The incidence of cervical cancer was very, very high. And it turns out that the vast majority of those cases were associated with HPV infections. Okay? Over 35 years, what's happened is that you can see the incidence of cervical cancer is plummeting. Okay? Now, of those cases that are there, 99% of them are still caused by HPV. Okay? So the question is, well, what accounts for the decline? Well, there's two. The first is behavioral modification. So people are having intercourse with having safe sex. And so what you're doing is you're reducing or mitigating the risk, just like any other STD, just by changing sexual practices. So the incidence of infection is dropping because you're reducing the spread of the virus. But from a clinical perspective, the other thing that has attenuated the increased rates of cervical cancer is the development of vaccines. Okay? This type of cancer has a viral etiology. We can stop viruses with vaccines. And it turns out there are two very potent vaccines that are out there that are available, Cevarix and Gardasil, that can basically block the 5 most prevalent substrains of HPV-16. That is the causative agent for cervical cancer. And so what you've seen is as people modified their behavior, the incidence of cervical cancer drops, but it's also because more and more young women are becoming vaccinated, okay, in their teens. Now, that's the good news, okay? So you've seen a marked decline in the incidence of cervical cancer, basically by just like we would treat any other viral-induced disease. But while this was going on, there's been an increase in oral and throat cancers. Okay, so the incidence of these cancers has been starting to go up. Okay? So what's going on? Well, it turns out that the increased incidence of these cancers is associated with HPV. Okay? So what you're doing is you're seeing a shift from one type of cancer to another with the same viral etiology. Okay? So the question is, well, what's causing this shift over the last 20 to 30 years? One type of cancer goes down, another type of cancer goes up. Well, there's three things that have come out of this. One is that the increased incidence of oral and throat cancers that are HPV associated are now affecting men to a greater extent than just women. So whereas cervical cancer obviously is completely female specific. So we've got a change in gender. The other side of it that was associated is that it's tied in with a change in sexual practices. People go away from unprotected intercourse, but they still have unprotected oral sex. And so the virus is being transmitted now, but it's coming into contact with other types of tissues. So there's a change. The other thing that is happening now, that more recent findings are coming out, the virus has mutated. And so the strains of HPV now that are more associated with oral and throat cancer is genetically different than the virus that was associated with cervical cancer, which means that the tissue specificity, the sequences and the viral proteins that bind to cell surface receptors are not the same now as they were 30 years ago. The virus is evolving. And this is something that you see with other viruses like Ebola. You see it with HIV. And so the problem is the virus itself is changing and evolving. So there's a couple of points that come out of this. The first is it's a preventable disease, and it's preventable by vaccination. So the one thing that this is calling out and emphasizing to even a greater extent is the absolute importance of young people getting vaccinated, irrespective if you are male or female. And that is, young teens need to be vaccinated against this virus before they become sexually active. Men and women. Everybody ought to be vaccinated against this virus. It is absolutely absurd that this type of cancer should go ahead and be as prevalent as it is in our population. Now, a lot of people will say, no, we can't do that because it will promote sexual promiscuity. Come on, you're stopping a disease, okay? The moral imperative of stopping a preventable infectious disease by vaccination would seem to trump any societal concerns about whether or not individuals who might be vaccinated are more or less likely to be sexually promiscuous. It's the same argument that was made 30 years ago against coming up with a vaccine or drugs to treat HIV. So the same arguments that were dragged out then that were disputed, basically discarded, they're now being used to argue against vaccinations against HPV, okay? So the point is, it is the one type of cancer that we can look at and we can treat it just like any other type of infectious disease. The other thing here to understand is that your p53 genes in these cells is still functional. It's still there. The gene hasn't been altered. The protein can still function. It's basically been hijacked by a viral protein. So now, as another way to treat these cancers is, again, to go in and try and find small molecules that will disrupt the interaction between the E6 protein and p53, to basically displace it. And again, the approach would be to treat these types of cancers is to allow the p53 that's already in these cells to restore its functionality and let it go about doing what it is good at, and that is causing the cells to commit apoptosis. So you're seeing this coincidence of basically trying to rescue or restore use p53's natural functions to basically treat different types of cancer. Okay? That's my public service address for this morning. Okay. So here's what we're going to move into now, to kind of look at the last bit of the unit in the course. What makes a cancerous cell cancerous and distinguish it from a normal cell, phenotypically. Okay? And the problem is it's a grayscale. It's not absolute. So when you say a cell is cancerous, what do you mean? Okay? There are certain hallmarks or there are certain properties that cancer cells have that you can sort of use as a benchmark and compare them to normal cells, but it's not absolute. Not every single cancer cell shares all of these properties, and the properties don't remain constant in one time. And what you're going to see is that any particular oncogene is insufficient to cause a perfectly normal cell to become a full-blown metastatic cancer cell. It's going to take multiple events, multiple oncogenes, and the loss of multiple tumor suppressor genes. But if we just take a look, okay? Oops, sorry guys. If you just take a look, if you were to define looking at cells in culture, so if you're taking tumor cells and growing them in culture and compare them to normal cells, there are some very, very obvious properties that you can see that distinguish them, okay? Now, one of the words that we use here to talk about cancer cells, we use this word transformed. Now, the problem with biologists is we use transformation to talk about all sorts of things. Like when we take plasmids and put them into bacteria, we call that transformation. When we take DNA and put it in any cell, we call that transformation. We're using the word transformed here in a different context. Transformed refers to a cancerous-like phenotype, okay? If you take a look at this, this is a dish of normal cells. And I put a star here, and this is something we're going to have to deal with. Is it, well, what do you mean by normal? What's the definition of normal? Well, what we're going to be looking at is that a normal cell is any cell that's not cancerous. Does that help? Okay? But if you take a look at these cells versus transformed cells, these are some characteristics that we can look at, that you can use to sort of size them up and look at the different phenotypic traits that they have. The first thing is normal cells exhibit a property called contact inhibition. And that is you take normal cells, put them in a petri dish, give them a growth media that's supplemented even with growth factors now. Every cocktail of growth factor, glucose, amino acids, you name it. The cells will grow to what's called confluency. And what it means is normal cells will grow in this dish until they're contacted on every side by their neighbors. And then they stop growing. And that's called confluency. And what it means is if you take a look at a petri dish that's grown with normal cells, you'll have a monolayer that's one cell layer thick. Every cell's touching another neighbor. Now, if you go in and you score the plate and you scrape away some of the cells and you create a wound, like we talked about in the cytoskeletal unit, that will cause the cells to re-enter the cell cycle and grow and migrate until they become contacted and inhibited again, and then they stop. Okay? Normal cells basically stop then in response to cell-cell interactions. Cancer cells or tumor cells don't do that. They keep growing and growing and growing, and they pile up on top of each other. They actually make what you might think, if you look at them, superficially they look like little small tumors or clumps. And these little clumps are called foci. If you have one of them, it's called a focus. If you have multiple, you call them foci. So tumor cells lack contact inhibition. They just grow on top of each other, pile up, and they make a mess of your petri dish. They're not all nice and confluent. Second point is, if you take normal cells and even trying to get them to grow to confluency, you have to add growth factors. The cells will not grow unless you give them a source of growth factors. Now, usually when you're growing cells in culture, as many of you have done when you work in a lab, you don't use a purified growth factor to make them grow. You treat them with serum. Okay? So serum is the liquid portion of plasma that contains soluble growth factors. It's indispensable for them to grow. And that's because, as you guys know, cells will arrest in G0 and remain quiescent unless they're exposed to a growth factor that activates that whole signaling pathway we went through to kick them out of G0 and to get them to divide. Okay? Cancer cells don't need it. Now that's kind of a no-brainer because, as you guys know, cancer cells are going to have acquired at least one or more oncogenes in RTK signaling pathways. And most of those mutations mean the cells no longer need a growth factor to grow. So tumor cells, that's one way you can distinguish them. You don't need to add extracellular growth factors. Yeah, they'll grow better with them, but they grow fine without them. Anchorage dependent. This ties in to metastasis. And that is, not only do normal cells exhibit contact inhibition, but look at their morphology. They're flat. They're spread out. Normal cells like to stick to a solid surface to grow. Okay? Now, normally in the lab, that's plastic. In our bodies, it's not plastic, but it's a coat of protein that's usually either collagen or fibronectin. But the point is, our cells only like growing on a solid surface. Cancer cells don't. Cancer cells are slippery, and they don't require this anchorage-dependent growth. These guys grow perfectly well piled up on top of each other. And what you notice is, you notice they have this rounded morphology? The reason is cancer cells secrete proteolytic enzymes that are disrupting and breaking down and digesting the interaction between integral membrane proteins and the solid surface on which they grow. If you take a dish full of tumor cells like you would have here, actually if you take normal cells, you take a dish and you shake it. It's like if you're walking down the hallway carrying your cells from one lab to another, you can shake them up, nothing happens. You do that with a dish of cancer cells, they'll detach. They'll be floating around. That is why these cells are more prone to metastasize. They don't stick in one place as well, so they're more readily dislodged and they can migrate somewhere else in the body. So the lack of anchorage dependent is also a property of these cells. Okay? Checkpoints, we've already been through that. These cells have DNA damage checkpoints. Cancer cells typically have lost them because P53 is gone. Okay? Karyotypic profile. Cancer cells often have chromosomal aberrations. Leukemias and lymphomas in particular are very prone to chromosomal translocations, like Burkitt's lymphoma or chronic myelogenous leukemia. Genes have moved around, you've got chromosomal abnormalities. So if you look at the karyotype, if you basically take a look at the chromosome profile in a cancer cell, it's not normal. They'll often have extra chromosomes, they'll have chromosomes that are lost, they're a mess. And now we get down to the kicker, this proliferative lifespan. Normal cells have a finite proliferative capability, with the exception of stem cells. Cancer cells can propagate indefinitely. Now, as you're going to see, some normal cells can too. But the vast majority of normal cells have a finite ability to proliferate. Cancer cells have a property of what we would call immortalized. As long as you take these cells, feed them, split them, take care of them, you can propagate them indefinitely. Okay? So, the outlier in the clear distinction between a transformed cell and a normal cell really comes down to this observation about immortality. So we need to spend a couple of minutes just talking about this. Okay? These experiments go back to some classical studies that were done in the 1960s. They go that far back by a scientist in Philadelphia named Leonard Hayflick. And Hayflick was studying human cells in culture and looking at their proliferative properties. And what Hayflick found was the following, is that if you take primary human cells and grow them in culture, these are non-transformed cells, if you take these cells and grow them in culture, okay, he was doing these studies at the Wistar Institute, so all of his cells were given the designator WI. So the particular strain he was studying of cells were WI38 cells, Wistar 38. These are normal human cells. You take these cells out and you start growing them in culture. And what you find is that they will go through about 50 generations, 50 cell cycles, 50 mitotic events or doublings. And then you notice what happens. They die. At about the 50th cell division, they check out of the cell cycle, and no matter what you do with them, you cannot get them to grow anymore. They enter senescence. And that is, they remain arrested in G0, and then they ultimately just die. It's as if there's a molecular clock in these cells that tells them you've got 50 cell divisions ahead of you, but when they're done, they are done. And here's what Hayflick did to prove that. He takes a population of cells and starts growing them in culture. He takes the cells at different times. Let's say after 10 doublings, 25 doublings, 40 doublings. You take the cells that have gone through that many cell divisions and you freeze them. You put them away in a cryoprotective agent and you freeze them down. They're in suspended hibernation. You take the cells that have gone through 10 doublings. You freeze them, you let them sit for days, weeks, months, years. Thaw them out, put them back into culture at 37 degrees. They start growing again. But they remember how many doublings they went through. And so if you take a cell that had gone through 10 doublings, thaw it, put it back into culture, and allow it to divide, it's got 40 left, and then it quits. Take a cell that had gone through 25, thaw it out. It'll do 25 more. Take one that's gone through 40, it will only go through 10. The cells know how many doublings they've undergone. So what it means is that the finite proliferative capacity is intrinsic to our cells. They know how much they can divide before they check out. In essence, our mortality is pre-programmed into most of our cells. It turned out that studies then that were done by other laboratories at the same time found that the reason for this was due to sequences that are found on the ends of our chromosomes that are called telomeres. And that is, telomeres are responsible, it's this unique structure at the ends of our chromosomes that's responsible for chromosome maintenance and replication. And every time that our cells divide, our chromosome telomeres get a little bit shorter and shorter and shorter. And after 50 doublings, they then basically become so short that the telomeric sequences are lost and the chromosome itself is lost, and it causes the cell to go senescent and die. So telomere shortening is found every cell that goes through this process of cell division with one exception, and that's stem cells. Stem cells can maintain the length and integrity of their telomeres. The reason for this is that in our normal cells, the enzyme that is responsible for replicating telomeres, it's an enzyme called telomerase. It's an RNA protein complex. Telomerase gets shut off in our cells. So most of our cells don't express telomerase. So over time, our telomeres get shorter and shorter and shorter. Now, is there some way that we can reverse this or restore it? Yeah, so if you go into cells that are becoming senescent and you reactivate telomerase, do the telomeres grow? Yeah, and so do the cells. They become cancerous. And the reason is, it's very, very difficult, if not impossible, to regulate telomerase activity. We're not at the point where we can control it that well. It's kind of an all or nothing enzyme. Either it doesn't work at all or it works too well. And if it works too well, telomerase activity causes our telomeres to become very, very large. We have even longer telomeres than we normally have, and that causes your chromosomes to become unstable and preferentially causes chromosome 8 that contains p53 to undergo a deletion. So, the good news of telomerase is that you need it for normal cell proliferation. Look at the choice. You have too little, you go senescent, you die. You have too much, you become a cancer cell. Now look at the difference between human cells and mouse cells. And this is just the point I want to finish with. Mouse cells, if you take them out of a fetal mouse and grow them up like you did the human cells, aren't the same. And this is why mouse cells are somewhat questionable to be used as a model for every aspect of understanding human cancer. If you take mouse cells and grow them in culture, what you find is the cells very rapidly start dividing. They quit dividing. They become senescence within a month. Most of them die very quickly. That's different than what our cells can do. But what happens here is after about a month, what happens is you look in this dish of cells where the vast majority of them are dead. And now what happens is a little small cohort of these mouse cells will start proliferating again. Something happened to these cells. They underwent a mutational event. But it occurs at a very high frequency in mouse cells, but not human cells. And what it now means is that these cells become immortalized. Now, they're not cancerous. And the reason they're not cancerous is you take these immortalized mouse cells and inject them into an animal, they do not cause cancer. You take cancer cells and put them into a mouse, they will cause cancer at a very high rate. So the thing to understand, and this is leading into what we'll get into on Monday then, is that it's these immortal mouse cells that are generally used to define normal cells. So these immortal cells that you use from mice basically are like normal cells with every regard: contact inhibition, confluency, anchorage dependent, they require growth factors, but they are immortal. Okay? Those are the cells that are being used as considered to be normal cells that were used as the test beds to isolate human oncogenes. So when we talk about isolating human oncogenes and looking for their ability to transform normal cells, understand that these are the normal cells that were being used for those experiments. Okay? All right. So that's where we'll pick it up on Monday to wrap it up then and take a look at how we're going to get these things back to normal again.